The human body is made up of trillions of cells. Within these cells are the building blocks that make us who we are, passed down from generation to generation. These building blocks are called nucleic acids, bound together in the form of deoxyribonucleic acids, DNA, and ribonucleic acids, RNA, both containing our essential genetic coding and key to the replication of all proteins and enzymes which support all human functions. Nucleic acid delivery has been revolutionary in its treatment of diseases caused by mutated or poorly controlled protein expressions, offering a higher proficiency and specificity for its target cells. This is achieved by the modulation of protein expression profiles of the cell by intervening with either gene transcription or translation. By doing so, nucleic acid-based drugs can increase the specificity of action and address the cause of the disease at its genetic level. For example, if we look at a disease such as hemophilia, where the functional protein is missing due to a gene mutation, this gene can be restored by the introduction of the correct gene or messenger RNA transcript back into the target cell through means of a viral and non-viral delivery vectors.
Experimental transfer of nucleic acids to eukaryotic cells has advanced our understanding of eukaryotic gene expression and regulation for over 50 years. In 1965, Veheri and Pagano described the first method of nucleic acid delivery known as the nucleic acid transfection technique. This technique involves diethylaminoethyl DEAE dextran-mediated cell transfection. The positively charged cationic DEAE dextran molecule binds to the negatively charged phosphate group of a nucleic acid, allowing it to enter the cytoplasm via endocytosis or osmotic shock induced by DMSO or glycerol. In 1973, Graham and Van der Ebb developed a new technique for assaying infectivity of endovirus 5 DNA using calcium phosphate precipitate as a non-viral means of delivery of nucleic acids. This resulted in up to 100-fold more plaques than could be obtained by using DEAE dextran. Newman and his colleagues introduced electroporation in 1982. In this technique, electrical currents are used as a means to facilitate DNA uptake, increasing cross-membrane transport. In 1987, Felgner and others described lipofection, a lipid-mediated DNA transfection procedure. This method involves cationic lipids forming smaller liposomes with positively charged surfaces. The liposomes have a distinct attraction to the phosphate groups of DNA, forming lipid DNA complexes with 100% entrapment of the DNA. This technique is simple, highly reproducible, and effective for both transient and stable expression of transfected DNA. Later, in 1998, Budka and his colleagues demonstrated that large volumes of plasmid DNA could be delivered to and expressed within skeletal muscle when injected rapidly in rats. The results of the experiment showed that intravascular injection increases expression of DNA in muscle up to 40-fold as compared with intramuscular injection in adult mammals. Intravenous delivery of plasmid DNA combined with cationic lipids enabled higher levels of foreign gene expression in the lung. However, these cationic DNA complexes cause substantial toxicity. In 2003, Chubaretskoy and others discovered that the inclusion of polyacrylic acid to these DNA cationic lipid complexes resulted in significant levels of lung expression being obtained while avoiding adverse effects. Nucleic acid-based drugs offer the potential to treat diseases by modifying the protein expression profiles of cells by interfering either with gene transcription or translation. This class of therapy offers high specificity and efficacy, as well as the potential to address the genetic causes of disease. There are many nucleic acid-based therapeutic approaches to altering gene expression. The most basic of those, classical gene therapy, increases the expression of a target gene by combining it into viral vectors or bacterial plasmids and expressing it in tissues to restore normal pathological expression. A wide variety of strategies using short oligonucleotides have emerged in recent times relating to the treatment of various diseases, including classical antisense oligonucleotides, splice-switching oligonucleotides, antigene oligonucleotides, small interfering RNA, microRNA, and anti-microRNA. Antisense oligonucleotides moderate gene expression by incorporating enzymatic degradation of targeted mRNA and the removal of the disease-causing gene product. Splice-switching oligonucleotides offer great potential for RNA targeting therapies. They moderate pre-mRNA splicing to restore functional protein isoforms and directly inhibit the toxic effects of pathogenic RNAs by binding to either the 5' or 3' splice junctions. This promotes an alternate use of exons, exon inclusion, or exon exclusion. Antigene oligonucleotides are designed to bind to double-stranded DNA via the formation of triple-stranded complexes and inhibit gene expression at the DNA level. They interfere with the initiation of transcription by binding to the transcription factors and stalling RNA polymerases. One of the most significant advances in RNA molecular biology has been the discovery of small, non-coding RNAs that regulate genes and genomes. Short interfering RNAs regulate RNA processing, chromatin structure, RNA stability, chromosome segregation, transcription, and translation. Double-stranded short interfering RNAs are useful tools in silencing gene expression. They are usually 19 to 22 base pair duplexes, which is a sufficient length to form a stable duplex and bind the machinery of the RNA-induced silencing complex, but short enough to avoid most of the strong interferon response provoked by duplexes greater than 30 base pairs long. MicroRNAs are short non-coding RNAs that moderate gene expression post-transcription. They typically bind to the 3' untranslated region of their target messenger RNAs and inhibit protein production by translation, silencing, and destabilizing the messenger RNA. To further control the regulation of messenger RNAs, anti-microRNA oligonucleotides neutralize microRNA function in cells for desired responses when overexpression occurs. There are two main strategies for nucleic acid delivery: viral and non-viral. Viral vectors are remarkably effective in delivering genetic material to cells as a result of evolution over millions of years shaping and optimizing them for this purpose. However, in some vectors, viruses will retain some of their inherent weaknesses, and as such, viral vectors are not universally applicable for all nucleic acid-based molecules, as they are incompatible with the delivery of short synthetic oligonucleotides. Non-viral delivery vectors can be based on many different classes of compounds, including various cationic lipids, polymers, peptides, or carbohydrate analogues. Another of these classes are cell-penetrating peptides, which are cationic or amphiphatic peptides with a remarkable ability to convey different cargo molecules into cells and tissues. Importantly, they demonstrate great potential for the delivery of nucleic acids ranging from natural or synthetic short RNA and DNA oligonucleotides to much larger plasmid molecules, both in vitro and in vivo.
Recent applications in disease therapy have gained significant benefits from the advances in the medicinal chemistry of nucleic acids. Gene therapy has provided modern medicine with new perspectives that were inconceivable 25 years ago. Recombinant DNA technology is commonly used to treat patients suffering from genetic disorders and also kill tumour cells in cancer patients. Various techniques of nucleic acid delivery have been developed for current treatments of diseases with differing characteristics and sizes. Physical transfection by electroporation is a non-viral vector technique where high-voltage electric impulses can be delivered into a neoplasm by transiently increasing cell membrane permeability to large molecules. This method can be used to treat large tumours. Liposome-mediated gene transfer is another non-viral gene transfer technique used to deliver radiosensitizing agents to tumour cells, which improves the therapeutic ratio of chemotherapy. Retroviruses are a form of viral vector gene therapy. These are RNA viruses that carry a gene for a reverse transcriptase that transcribes the viral genetic material into a double-stranded DNA intermediate. This is then incorporated into the host DNA, allowing the host cell machinery to produce all the necessary viral components. Retroviruses and other viral vectors are used in cancer treatments and vaccine production.
The delivery of nucleic acids has the potential for prevention and treatment of cancer throughout the body, liver disease, neurodegenerative disease, gastrointestinal disease, and dermatological disease. Non-viral vectors are much cheaper and easier to produce in large amounts. They have limited immunogenicity, which allows for redosing. They are also considered safe, and there is no possibility of recombination that would result in a competent virus that could potentially cause disease. Viral vectors have a more favorable system of gene delivery. Gene transfer is more efficient and specific than physical and chemical methods, and a single dose is generally sufficient. While there are many advantages and uses of nucleic acid delivery for disease therapy, the technique has some disadvantages in the delivery itself. In order for nucleic acids to be used in vivo for disease therapy, they must reach the cytosol or nucleus of the cell within the target tissue. This presents two main challenges. The nucleic acid must limit exposure to non-target tissue, and it must be transported to the intracellular compartment where it is required.
We have only reached the tip of the iceberg when it comes to potential use of nucleic acid delivery for disease therapy. The ability to modify and correct defective genes could lead to tremendous achievements for the medical world and have the potential to prevent genetic diseases such as cystic fibrosis, sickle cell anemia, Huntington's disease, and numerous forms of cancers. With such potential, it is no surprise that nucleic acid delivery has been such a high study priority among the scientific community. Some of these studies include the delivery of therapeutic oligonucleotides. More specific, the use of oligonucleotides to combat diseases affecting the central nervous system. For a long time, researchers have been stumped at a crossroads when it came to accessing the central nervous system compartment, due to difficulty passing the blood-brain barrier. The blood-brain barrier contains tightly connected endothelial cells secured by a network of pericytes and astrocyte processes. This structure is largely impermeable to oligonucleotides, resulting in limited success efforts for delivery into the central nervous system in the past. However, researchers have recently overcome such hurdles with the direct administration of oligonucleotides through intrathecal injection, which has allowed the introduction of oligonucleotides through the central nervous system, resulting in direct uptake by neurons and glial cells. This breakthrough has opened up the door for possible treatments of many other diseases, and is just one of many achievements in the promising realm of future applications and practices for nucleic acid therapy.